Okay, good morning, everyone. Welcome to the last day of the particle size measurement training course. So again, as I mentioned this morning, we will talk about the method validation, and also I'll share with you some of the deficiencies that we have been observing. At least we were part of the review team that we have been providing some of the feedback to the review teams. And so I picked a few examples related to dynamic light scattering and laser diffraction. Those are the two most commonly used techniques that we have seen in application. So I picked a few examples just to help summarize some of the things we have learned in the last few days. So beginning of that discussion, so I will try to go over some of the general considerations, and this usually are for the developer, so basically the applicant. Oops, it advanced automatically. But I think it's equally relevant for you as assessors, because what the difficulty that the developer typically encounter, I think it's good that you are also aware that you can take into consideration when you are looking at typically it's the report or the data submitted by the applicant, and you're trying to figure out maybe what was the problem, or is this really believable, or is something that you want to ask a question, for example. So hopefully this kind of recap summarizes some of the things we have already went through the last 2 days. And this is not a complete list. So the first thing, of course, whenever you are looking at a technique, is what's important is to determine is it appropriate for the size range of the particle that we're analyzing, suitable, or the technique, is it suitable for analyzing the particles in that size range? And there are some clear, I think, kind of separations or ranges. So if the particles are really small, when we are talking about the sub 100 nanometer range, and typically the most reliable technique is the dynamic light scattering, because it's easy to perform, and also it deals with the Brownian motion of the particles in solution, and that's the one give most reliable results in the sub 100 nanometer range. On the other hand, the laser diffraction, as we talk about, it's analyzing angular dependent of light scattering, and typically more suitable for relatively larger particles, considering that it need to be at or above wavelengths of the light. So typically most reliable is when analyzing particle that's slightly larger than 1 micron. Just bear with me. I think I have my slide as automatic, but let me just disable it. Okay, let me share again. Sorry about that. Can you see the screen again? Hope that's yes. Okay, so the larger particles, we know the laser diffraction above 1 micron is suitable, but then there are situations where you may encounter particles that are highly polydispersed. So it's known to be a mixture of multiple population of particles. And if your desire is to know precisely what are the populations, the particles in there, and but possibly, if you want to know both the excipient, maybe our particles, as well as the particles of API or particles of drug in certain carriers, both you are interested. So now you have a different population of particles that you want to separate. You want to know what's the size range for each of them. Maybe you want to resolve them. So in that case, ensemble technique like dynamic light scattering or laser diffraction may be difficult to accomplish that goal, because they provide you information with non-specific what is the particle. So in that case, a separation technique may be beneficial to be applied before utilization of the light scattering phenomenon to analyze size. So this is what we typically consider as hyphenated technique, like what Hayo has talked about yesterday, AF4 or FFF, field flow fractionation based technique. So typically those are hyphenated technique, meaning they are used at the beginning of the analysis to separate the particles first. and then followed by a different detector. So can be a DLS detector analyzing hydrodynamic radius, or can be a multi-angle light scattering detector to analyzing molecular weight or other properties. So those techniques can be useful if you are interested in separating to know what is in this population and exactly what's the smaller portion and what's the larger portion, and what's relative composition, and also what's their impact potentially overall contributing to the size distribution. So in those cases, you might want to consider a separation technique. Of course, as a reviewer, you cannot dictate what the company use, but in case, for example, you are part of R&D, if there is a submission of R&D and the company raises a question, this is what we have, this is our product design. And then there are situations I've seen that there may be an opportunity for us to recommend them a certain technique that may be more beneficial to understand the product better, and in order to develop more appropriate product control strategies to ensure quality. So for that reason, I do think this is an important consideration even for you as a reviewer to recognize what is the range of the size that we are dealing with, and what is a more appropriate technique to analyze the particles in those size range. And also, like I think we mentioned the first day, second day, is that oftentimes the shape of the particles can be important if that's something you are really interested to know, like how does it impact the laser diffraction results if it's a really like needle-shaped particles impacting the scattering diffraction pattern, or what is the particles? Is it oil, or is this a glass, or is it aggregate? You might want to see the shape, the morphology of the particles by measuring, perform some sort of image analysis. So in this case, if the question coming to mind, not only I want to know the size, again, size may not be always important, but now if I want to know the shape and then consider recommending or looking for microscopy data, some of the image data could be, if the particle is relatively larger than the typical microscope, that it will be sufficient, can analyze particle down to 1 micron, 2 micron. The typical microscope is enough. But if you're dealing with particles that are much smaller in the nano size range, then certainly transmission or scanning electron microscope would be more appropriate. So again, if the information about the shape of the particle, morphology of the particles coming to picture become important that you want to know, you need to know, then the microscopy imaging-based technique is something that you can consider to recommend, or look for the information as a complementary information to allow you to assess overall the product. Also, keep in mind that keep asking this question: is a particle, is a sample sensitive to dilution? And this particularly is important for liquid-based dosage forms that supplied in either concentrated form or maybe a lyophilized product for reconstitution. Now, before use, the product need to be diluted to a certain way, and then what's in the finished product may be different than what's given to the patient. So it's important that we also keep in mind that this effect of dilution is something we need to consider. And certainly, I think we mentioned heavily in dynamic light scattering as well as laser diffraction, the impact of dilution in terms on the measurement due to the concentration. How does it impact the multi-scattering in case of both dynamic light scattering and laser diffraction, and how that impact the results of the analysis, right? So in that case, it's really good to know how the particles behave in different dilution conditions. Sometimes they will dissolve if the drug or if the particle has certain has enough solubility in the medium, in the dispersant, and then what you will find is a particle will disappear, or distribution will change or shift, or sometimes particles will aggregate, which is a different type of problem than dissolution, right? So, and then next is whether or not the sample disperse well and remain stable in the medium. I hope you remember its first lecture, we talked about About the particles, I mentioned that it's important equally consider the medium, dispersion medium, equally as what the particle we're analyzing. The reason is particles do not exist in the absence of an environment in the medium. So it's really two equal important part. And of course, we typically pay attention to the particles. But what I want to emphasize here is equally important. We look at the dispersion medium, and how we look at that is by understand whether or not the change of the environment from air, for example, for powder material to liquid, how does it change the stability of the particles? If we have the suspension to begin with was suspended in sodium chloride, normal saline, if we dilute it using water, what does it happen? What does it impact the stability of those particles? So for that reason, it's good, it's useful to consider the stability, the dispersion, the stability within that dispersion medium. And also, this is one thing that I think a picture worth 1,000 words. Oftentimes, you might not be able to actually see what the product looks like. So it will be very useful if you either look for the pictures, look for the information provided by the applicant, or request such information that allow you to actually see what is the product and what does it look like. Because sometimes by just looking at the product may give you a lot of clue about what may be the problem. So one typical, what we have seen really easy to understand problem is if the sample is a really, really dark color, like black, a black dispersion. And this example, of course, iron colloid. We know iron is a dark color, absorb a lot of light. So if you have this iron colloid dispersion nanoparticles, and if you put into the path of the laser light, and most of the light will be absorbed into the sample rather than being scattered. So now, but we analyze the size, we analyze the particles by measuring what's being scattered. And because the sample absorbs so much light, really doesn't allow you to actually see the scattering signal from the particles. So therefore, if the sample is highly dark colored, and then this is going to be inherently, you should recognize is a challenge to measure that particle. So for that reason, if you don't see the picture, for example, then you really don't know what could be the problem. And the company may not even recognize that's being a problem, even though they should. So yeah, keep in mind, even the picture of the product, how does it look like, can be useful sometimes, especially when analyzing the size of the particles. This absorption is something very useful to know. Does the sample preparation impact the stability of particles? So similar to the medium, what I've talked about already, whenever we change the environment of the particles, whether it's diluting it or putting the sample into the measurement unit, a dispersion cell, we're changing the environment of the particle, therefore may have an impact on how the particle will remain in that state, whether it's stable. So typically when we are looking at larger particles, it's whether they will settle down. So certainly by increasing the stirring speed, can overcome that problem, or if the particle, the aggregate, then by sonicating it, you can keep the particles from coming too close to each other, and then to remain a good dispersion, which allow the measurement. So again, this preparation procedure is something that oftentimes you do not see, but it's something actually you can find out if you go to the procedure portion of the method, and then to look for that. But it's good to keep in mind that the sample preparation, especially again, changing the environment, changing the medium, has a really significant impact on how we can actually measure the particles. So it's not trivial by looking at the procedure portion of how the materials are prepared. And always ask yourself, does that procedure change the environment? If it does, what will be the information you want to look for to mitigate that potential concern? And does the formulation component interfere with analysis? I think we have seen some examples yesterday. We will see more examples today about how as inactive as we call them, inactive ingredient or excipient, they are actually a big part of the product and formulation that they play a really significant role also in how we can measure the particle. Typically is the drug portion, API. We're interested, but they are there to potentially interfere. So it's not something we can just ignore, consider them being inactive. They are inactive only from a, I think, pharmacological or therapeutic point of view, but they are certainly functional when you consider how they make this product, right? So, but if we look at on this slide, I mentioned quite a few of considerations. Many of them actually falls into some of the studies that during the first day I've been talking about, as part of the validation, or maybe I didn't mention as in the context of validation, but I did mention the context of how do we measure precisely of something, right? Precision versus trueness. Precision is much easier to achieve, right? But in terms of the precision, what we are really talking about are 3 R's: repeatability, reproducibility, and robustness. So if you look at what's on this slide, actually all of them, almost all of them, cover the 3 R's. Whether it's stability of the sample, which we can find out if there's a problem by looking at repeatability measurement, by measuring multiple times of the same thing, just a function of time, we see, does it change? If it does change, that indicates sample stability issue. Or is it robustness? Whether or not changing slightly condition, adding sonication, dilution, filtration, changing the temperature does impact the results or not. All of those can be part of the robustness evaluation. And reproducibility allows us to see whether the procedure, like preparing the material differently or by different person, does it introduce variability or not? So what the considerations I've mentioned on this slide, all of them almost can be covered, or the risk can be mitigated by a properly designed validation study of the particle size measurement, right? So it's something that you need to look for when you are analyzing or assess the application's data. And it's something that really you can just remember 3 R's. It's really useful. Okay, so now just to continue, the second thing you can consider beyond the 3 R's is what are some of the settings. So I know this can get to too much detail, but remember a measurement is not complete without knowing what are all the detailed conditions. So we talk about the diamond light scattering, laser diffraction, of course, AF4, and also imaging analysis for subvisible particles. But one critical part of any measurement is the settings in the software. And typically you do not necessarily see, but it's important for the company applicant to report. So in the case of dynamic light scattering, what's the measurement position? What's the laser power? Because that impacted the amount of light going into the material, amount of scattering volume, and amount of light actually will be scattered. So these 2 parameters together will adjust the concentration, compensate for the concentration differences of the material, and allow an operator to successfully complete one measurement. But by adjusting these 2 parameters equally, you can actually make the 2 material that looks actually different, or they are different, to look the same by adjusting those 2 parameters. So it's really important that you know how do those 2 parameters or few parameters impact the results. Certainly there's a diffraction, how fast do you speed, you can help improve the dispersion, or apply sonication, which will help preventing the particle from agglomerating together and maintaining a separation. Remember, there's example Billy yesterday talked about the particles over repeatability measurement, the size is getting larger and larger over the first 3 runs. And that's typically because of the agglomeration of the particles. And to overcome that, if you want to measure the primary particles, apply certain sonication will prevent the particles from agglomerating. And also optical input are really important, both techniques, scattering technique, because scattering optical input is such a critical component of the scattering phenomenon that without those information, we cannot necessarily perform a measurement. And we talked about the analysis algorithm for the dynamic light scattering. Just remember, there are 2 ways to analyze DLS results. One is cumulant analysis. So what that data provides you is Z average and PDI. So those are assuming sample population is monomodal. And the second way to analyze DLS data is by analyzing distribution. So applying certain level of regularization, assuming multiple distribution, and then so that give you what that gives you is a D10, D50, D90. So it's either Z average PDI or D10, D50, D90. Those are two different ways to analyze the DLS data. And of course, in the laser diffraction, we talk about Mie theory and Fraunhofer theory, which apply to different size range of the particle and different shape, etc. Lastly, but not least, this is what's really important, especially for you, is what should be reported in the applicant's application, right? So the short answer is the more the better, and as detailed as the better. What is a mean value, right? We talk about even the mean, we can describe using different way to describe the mean. Can be arithmetic mean, can be a volume weighted, can be surface area weighted, can be intensity harmonic means like the average. So what exactly is that number that the applicant provide to you? If they just gave you a number without telling you what is that number, is that a mean, is that a median, what type of mean? Really, there's a meaningless to see that number because that number can mean anything, right? And also, we talk about distribution, dispersity index, span are two ways to analyze the spread of a distribution. But PDI or polydispersity index, keep in mind, it's only suitable for DLS. It's only suitable for cumulative analysis in DLS. It's not applicable for distribution analysis, for example. Rather, for distribution analysis, it is a span value that's more critical. Cumulative distribution, we already talked about it. And last, anything has to do with how the data is generated, the settings, the preparation procedure, you should know more, and company should provide more. It's something that we have been telling the company. I mean, we meaning through different ways, through standard development organization, through public presentations. We have been keep telling the industry, provide more. But I know still in submission, sometimes you do not find all those information. So when in doubt, it's good to know what are the detailed procedure and condition. Those can really become helpful. Useful for you to actually diagnose or thinking about what might be a problem or there's no problem, for example. But without enough information, I think it's really difficult for you to make the proper appropriate decision. Sensitivity and specificity, so this is important as part of the recognizing that why we measure particle size is to use the information to allow us to make a decision to know what does the particle do, whether it's dissolution, process capability. So for that reason, if we want to use a size to achieve that purpose, we need to make sure this methodology can allow us to see any changes in the process, formulation, in the absence, of course, any interferences. And also, this point about the single technique versus complementary, I don't think it's necessary to require the applicant to do that because they may typically consider for QC purposes, for example, just using one technique, and only that's enough. But what's important, or where may become useful of using complementary techniques is during the product development to understand their product a little bit better, understand what they are actually measuring. And this is where the complementary techniques can become really helpful by, for example, dynamic light scattering, laser diffraction, electron microscope if it's really small, or just a microscope technique if it's large. So different techniques to provide information about the particle that we're analyzing. I hope you remember the story I told you about the elephant and the 6 blind men, where in our laboratory, we actually truly observe these differences in just by measuring different technique, we get totally different answers. So, but in the end, we were able to understand what is happening. So that really helped us tremendously. So the same token can be, same thing can happen for the applicant as they are developing their product. If they do use a complementary technique, that can help strengthen the understanding. But certainly, we would not want to ask them to use complementary technique to do product control, quality control. That would be too high of a burden, right? So that gets into the method of validation. What exactly is useful to be validated for particle size measurement? I know at FDA we talk about validation many times. Is one thing that we whenever we look at a methodology, we consider appropriate validation, demonstrate it's suitable for its intended purpose. But the question is, what's suitable for its intended purpose for the size measurement? You all know the ICHQ2 R1 and also USP 1220, I think 1225. There's a general guideline about what to perform as part of a method validation as applied to any analytical procedure. And typically, as you can see along these different rows, we have specificity, linearity, range, accuracy, ILD, ILQ, precision, robustness, right? System suitability as well. So typically, as part of validation, this is a complete list of the things we want to see, right? But keep in mind for the size, what's suitable for its intended purpose, not every of those parameters is suitable. So for example, we go one by one, specificity. If you consider DOS data, just to think about the histogram or the z-average value, PDF value that you received as part of the report. You know what's the range of the particles, but do you know what is a particle being measured? In a way, do you know the chemical composition of that particle? Think about if you have two particles, API and excipient, of exactly the same size, hypothetically, can you tell them apart by looking at just the size value report histogram? And the answer is no. Most of the size measurement is non-specific. We know what's the dimension, what's the size, what's the attribute we're measuring, but we don't know what they are. And we can make a guess of what they are, but we don't know what they are. So in a way, the method itself is not specific enough for us to distinguish one component from the other component. And for that reason, validation of specificity is really difficult or challenging and or not relevant. Same thing with linearity. Well, not the same thing, but when we talk about for the DLS and the laser diffraction, for example, it's a first principle measurement. The measurement is relying on the mathematical equations and is relying on the sensor reporting out some data and using that data, putting to the equations to perform a calculation. And the calculation do not take into account any other information other than what you put into the software. So in a way, you do not need to calibrate it. So there's no range of the size being valid or linear response to size. Almost I can say the most of the response in this particle size range are nonlinear. The scattering signal are nonlinear to the size itself. So this validation of linearity also it's not relevant. And that same goes to the range because the range it depends on what you are measuring. And typically is predefined by the technique that you are using. If it's DLS, then we know the lower end limit is about a nanometer around that range, and the upper limit maybe up to a micron, but more reliably for myself, I'm more comfortable. If it's a few hundred nanometer, I'm still comfortable. But if it's a 2 micron particles, I'm not comfortable using DLS. So the range of the size measurement is predefined by the choice of technique, not by the method that the company is developing. If the company is choosing one equipment, the moment that equipment is selected, the range is defined. The next one is a little bit tricky, the accuracy. As I mentioned earlier, the accuracy speak about the relative closeness of the measurement agreement with the observation true value. So trueness of in the sense of the size measurement is really difficult a concept to comprehend because what is true, right? But granted, I know many of you have seen applicant do this, and we reviewer do ask applicant to do is try a polystyrene standard, try a NIST size standard making sure you can measure that size accurately, right? But what we are truly asking the company to do or expecting them to do is not necessarily the accuracy. What we are expecting them to show is the qualification of their system to ensure, to demonstrate that every day when they're doing that, the system is functioning properly. So even with the use of the size standard, the purpose is not to ensure accuracy of the measurement, but rather to ensure the system is qualified to perform subsequent measurement. Think about that. If you measure 100 nanometer polystyrene uh, by dynamic light scattering, what do you need to put into, or not dynamic light scattering, let's talk about the laser diffraction. You will put into the optical property of the material, you will put into all the settings in the software, and then you choose the algorithm. What it generates is the size value. The question is, if you can measure a polystyrene bead of 2 micron accurately, exactly getting the 2 micron value that is matching what's in the label of that vial of the size standard, do you have confidence about the sample? Does it automatically mean the method that an applicant is going to measure their sample is going to be accurate? And I can tell you those two things are not related at all. The what you consider as is it the value I can trust or not of the sample really depends on the settings, the procedures, the sample preparation procedure going to preparing that material of the actual sample and putting the input of information into the software to perform that sample measurement. What the applicant did for the standard has nothing to do with the accuracy of the measurement of the actual sample. For that reason, this is a really challenging, I know, a part for the validation. I'm not asking you to not look at the, the size value measured by measuring the size standard. But what I want you to consider is, do not over emphasize the importance of the accuracy of that measurement. Recognize that measurement only give you confidence, give the applicant confidence that the instrument on that day is performing, it's operating properly. But that's all it can provide. It cannot tell you anything about the accuracy. In my years of experience in the lab, operating the dynamic light scattering and laser diffraction, I've not seen the instrument fail in a single situation. So instrument is actually being designed very robust. I'm trusting that if the company is doing that qualification every day, very unlikely they will see out of spec due to that instrument issue. So this is something that keep in mind. It's something you can look at, but you don't need to overly emphasize on the accuracy part. Certainly, if company do not perform the measurement using the size standard, it's not something you have to request them to do. Certainly, if they provide, that's good. You can look at it. And detection limit, quantitation limit goes with a linearity or goes with a range that because we don't, it's not relevant for the range and linearity. So therefore, really, there's no detection limit or quantitation limit. It's really the range that what is the size range that we feel comfortable the technique that can provide a measurement. So right, so far everything what I talk about is may not be relevant, but what's really critical is the precision, and the precision in terms of 3 R's: repeatability, reproducibility, and robustness. Of course, robustness itself is one of the critical things to validate. So that covers these 2 parts are both important, really important, right? The first slide I talk about so many things you can consider, and almost you can address all of those things by looking at the repeatability and the reproducibility and the robustness measurement. In a sense, a properly designed validation can address all of those concerns, right? So again, what is repeatability, reproducibility? I don't think I need to go through them again, right? Repeatability, you can test whether machine has problem or if the sample is stable or not. And the reproducibility, mostly testing the procedure of the preparation, sampling procedure, dispersion, and the operator to operator, error, human error, machine, and the robustness really is making sure that when we are expecting a change, expecting a signal, we do see the change. When we don't expect the change. We don't want the noise to become so significant that it causes a variation by overshadowing the actual signal, right? So these three things are really important to validate, and there can be a lot of things that need to be done as part of validation. So with that, I will ask, I guess, the first question. Let me bring my chat. And if you can provide A, B, C, D, E, what is the questions? C, 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 precision, robustness. Thank you. Everyone is listening. Everyone is listening. I'm giving a few seconds. It's a trick question. Hope someone noticed something else. So far, no. It's multiple choice. Yes, you got it. There's another. And of course, yes, C and E. Robustness, again, robustness is really important. And the robustness can go with a different form. You know, what you consider as robust, whether it's temperature, stirring speed, amount of sample that's added, the position of the sensor, the effect of procedure, diluting. And so all of that, what we want to make sure is that company actually know what they are doing, and what they are doing is not going to cause misleading results, right? When you are looking at the report, you're trusting the company generating the data reliably, and that data is you use as a basis for you to make decision. Can I approve it? Is this acceptable? Is this limit acceptable? Is this data supporting what I need to make the decision on the adequacy of the method? So robustness, it's really, really critical. And of course, robustness is part of the precision that, as I mentioned, 3 R's, and all of them, all three are important. So thank you. Glad no one mentioned about accuracy. I have been emphasizing quite a long time, I know, repeating myself on the accuracy. Really difficult to determine what is accurate, what is true. Okay, so the next, I will quickly go through some of the deficiencies that we have seen. And I put a disclaimer, I've not seen, well, I'm not actively searching for reviews, searching for deficiencies. So the one I'm providing here are the one that at least we have been involved in application through consult. So we will ask, hey, can you look at the method? Can you provide us some input? So this example that I'm providing is the one we did. And I find a few trends. Certainly you will see a lot of example. I'm talking about a globule. So I can tell you this has come from the cyclosporine ophthalmic emulsions or difluprednate ophthalmic emulsions. We recently just had the first approval for both of them, first generic for both of them. One cyclosporine, 20 years since the original ND approval. So one of the things we did is helping really understand how to perform this type of measurement on the globule size. And because of globule size become really critical component of the bioequivalence demonstration. So with that disclaimer, this may not be complete. I know you might see a better example that feel free to share. I'm really interested to hear from you if you have another example that you can share that matches what we have been discussing in this course so far about some of the key considerations that you think really resonate well with you, then yeah, definitely let us know some of the examples. And also if you have an application that you really want some of the input and feedback, yeah, also let us know so that we can give it more kind of try, and then maybe we will share some of the deficiency in the future with other reviewers. So this first example is about how the refractive index value is used in a dynamic light scattering measurement, right? So in this case, the company used a RI value of 1.52 for the castor oil, which is the oil globule, right? It's the actual particle you can consider. And if you remember, when we mentioned about the globule size or particle size measurement, what's important for the refractive index is the refractive index of the dispersant, right? It's because the dispersant RI directly is in the equation of the equation to calculate the hydrodynamic radius. But why do we ask here the Ri value of the particle, right? You may consider, we have been telling, it's not important to consider Ri. What's important really is dispersion, but why do we ask here, hey, your Ri value is not correct? The reason is this applicant, they did not use cumulant analysis for setting their controls or acceptance limit. Rather, they are using D10, D50, and D90. So what is D10, D50, D90? If you remember for the DLS, those are distribution analysis. And distribution analysis does not assume the monomodal distribution. Also, distribution analysis utilize complex, regularizer setting, etc. And we also don't know whether they are reporting is intensity, volume, number, right? So with that unknown, if they are using cumulant analysis, Z average, then this question will be completely not relevant. But because they are using distribution analysis and there's this uncertainty about what the value they are providing, and it's important to use correct, or asking them to pay attention to the correct input value for the refractive index. And that's the reason there's this question about RI. And we have seen many kind of common problems. And typically it's around, a mixture of did they use correct RI for the dispersion? Did they use cumulant or distribution analysis? And typically when the two mix, it's where confusion occurs and is where problem occurs. So just keep in mind that RI typically for the size analysis, if based on the cumulant, the first principle direct analysis, you do not need to know the RI value. But if you are using distribution analysis, which involve much more complex analysis calculations, then it's better the applicant know what they are actually doing. Because in that case, imagine by changing the RI value from 1.52 to 1.47, which is correct value, they may changing their product from out of spec to within spec, right? That's definitely, we don't want something to happen that the applicant knows this thing is something they can change later, and that without you knowing it, the quality being compromised. So that's the reason we want to know, because it's the type of analysis being using. The second example, also tied to the what type of results analysis they are using. As we mentioned for the dynamic light scattering, there is cumulant analysis and there is this distribution analysis. And when the company is using D10, D50, D90, that clearly tells you they are using distribution analysis. Now the question for the distribution analysis, secondary question is, what is the weighted function they're using, reporting that D10, D50, D90? Is that intensity-based, which is raw data, which is relatively safer to use? Or is it volume-weighted, which is carry much higher risk? Certainly we do not, I think we shouldn't allow use of the number-weighted number because that's totally can be misleading. So, right, because here we do not know what exactly is the weighted value they're reporting, that we need to ask them, clarify what exactly is the value they're reporting. And also, in the application, I searched the beginning to the end, I couldn't find what is the algorithm they're choosing. They're using Marvin zeta sizer. So we know that's one of the input or parameters. So we ask exactly, general purpose, multi-narrow mode. This is exactly the Marvin software setting. And they're supposed to be easily answer that question, because this answer important is it tells you whether or not they're using different regularizer setting. And if they're not telling you, that means they can change later, right? If they are not specified anywhere, and then if they make that change, it's completely to the operator. They can change that setting on their own. So there carries the risk of not knowing, because it's something that really can make an impact. Again, we're talking about out of spec, within spec, whether or not during the shelf life, after you approve the product, is the product still the same from the day that is approved? The next example is about viscosity. And I think this is perhaps one of the most critical input parameter in the calculation of the hydrodynamic radius in dynamic light scattering. And many mistakes can happen, or many unfortunate mistakes can happen if the input value is not correct, because viscosity, if you remember the Stokes-Einstein equation, I hope you have some memory, that viscosity is in the denominator of the equation. So if you, let's say, instead of 1, you put 2, right? So what that means should be 1 centipoise, but if you put 2 centipoise, and your particle size will automatically be deducted by half. So, so it's direct relationship between the input value of the viscosity and the output of the size value. So in this case, their value, they are using incorrect dispersion viscosity for the temperature that they are using. So we're asking them to use the correct value. And this is more of, I think, good to know. If you considered everything else is okay, and only this input parameter, I would say it's something you can make your own judgment whether it's really necessary for them to correct the number. So what this does is, instead of 100 nanometer as an output, maybe they were generating a value that's 110 nanometer as an output. But if they're comparing their product to another product, or batch to another batch, or to reference to a test, may not make too much a difference if you are just for the sake of comparison. Or if they set their spec based on the 100 nanometer, they set, let's say, from 80 to 120, then the fact that shifting to 110 also may not make too much a difference. So if everything else, in your application looks really approvable, and this may not be an issue that you have to get them to correct, but there's always an ideal way to do things, and ideally, the size value, you want to use the correct input to calculate the size value. So the next is related to how the concentration choices, and which is related to, in this case, this is a dynamic light scattering, sorry, which has a measuring position and attenuator setting. Remember, these two parameters can be adjusted and to compensate for different concentrations. So if the goal for the applicant is to compare product to product, really it's better for them to fix the condition to compare. Otherwise, you may have two products show the same size, but actually they may have different concentration because one product has more process loss. So if they fix the measurement condition, position, attenuator setting, that concentration effect will show up as a difference, but otherwise you were just measuring the size value. Okay, let me speed up a little bit to hopefully finish that sooner. This is about how to set a specification. What I typically find is applicants set a specification really, really wide, almost you can fit a truck through it. It doesn't really matter what's their product. It'll all pass. So a typical, if it's a generic application, of course, the good reference is the reference product and using the 3 sigma to set a spec, or they can use their variability to set a spec, right, based on the multiple batches, multiple measurement, based on the variation and the set spec. So this is something I think you're all very good at, so I'm not going to comment too much on. And what is need to be validated, right? So repeatability, reproducibility, robustness is really important. And I think these are two kind of applications. Those are not from the one application, there's from two applications, but share very similar thing, that basically asking them, you are using this dilution condition, but do you know if you change the dilution, does that dilution condition change your size value? So why don't you perform a robustness study to evaluate the impact of dilution on the size value? So make us confident that when even you make slight changes, even there is an operator error in the dilution, we will not be worried about the value that's produced. So still can be used to release a batch, for example, or demonstrating equivalence, another example. How to identify the problem. So this is related to the variability. And a lot of time we see variability is due to the method not properly developed. And when the method is not properly developed, sometimes it's due to the interferences. I think we have one more example later. So that's all some of the kind of what we have seen are the common deficiencies. Again, may not cover all you have seen, and you may see more. So please share with us when you have a good example. And next is not a deficiency, but it's a question that raised by the applicant as part of the pre-ANDA process that we were able to address during that pre-ANDA timeframe. And I think Billy already talked about it. This is a suspension product. Applicant stating we have two peaks, but how do we do the population bioequivalence? How do we compare the generic product to the reference product? Do we need to compare two peaks, or the two peaks, the second peak is really not important? Can we just mathematically drop the second peak? So basically, internally, we did some work to find out actually this secondary peak is not what, like when I said non-specific, is that the second peak actually is not the product we're interested. Rather, this is interferences from a polymer, carbomer. And through some of the method development, we can actually eliminate the interferences, which allow us to really analyze the particle of interest, in this case, the API particles. So certainly we have published this method, and we hope the applicant also may find it useful and they can start using. But this is something that by interacting with review, joining the review team, we have been able to learn over time. So again, I really appreciate if in the future we also have more opportunities to work with you. If you find something, let us know that we can accumulating some of the understanding and generating some results. So here's another example about the Billy also talked about it, which is one of the other case applicants say we have two peaks, but we only think there should be one, and the other one just ignore it. But can you ignore it? What's the reason for the other peak being generated, right? What's the root cause? And here there are information that can provide that allow you to understand better. On the left, this is concentration as a function of time, and the bottom is as a function of dilution. What's the measured the volume weighted size value D43, and those give you clue the particles are dissolving. Right. And then also, if we look at time course of the histogram, you see that one peak becomes smaller, the other peak appearing. That clearly indicates a shift of the particle population. And in this case, we understood, and this is brinzolamide and also Lotimax, it's the same. Those two products share a very similar situation, like what's shown on this slide, is that both lotipredniol suspension and the brinzolamide suspension, they all have this problem of potentially drug dissolving during the measurement. So need to really saturate the drug solution and also choose the appropriate setting, medium, etc. Other common deficiencies, I think I've gone through all of them already, so I probably don't need to repeat importance about the choice of the technique, RI, whether it's cumulant distribution, what is weighted function, and also, at this point, I probably have not mentioned, emphasized enough that validation is really important using the actual testing material, right? Using the NIST size standard, like I mentioned, for the accuracy part, cannot tell you anything about the sample that they're about to measure. Those are completely separate. An outcome of the measurement from a size measurement depends on the procedure, depending on the setting. And for each type of material, you need to use a different setting. So when you use one setting for the size standard, the other setting for the actual sample, there's nothing that you can compare. You're comparing apple and oranges. So really validation is really critical to perform the validation using the actual sample, whether it's a reference product, if it's generic application, or it's if it's a brand name product, just using exhibit batches or trial batches. But it's important to use the actual sample material. Lack of sample method detail, missing critical information, all of these things are something you should keep in mind. So with that, I want to close this morning session, the first session, by asking another question. What material is more critical to be used for particle size method validation? I think I just gave you the answer a minute ago. Yes, it's actual sample. It's much more valuable than using a reference size material. It's not that reference standard is not useful. It is useful for qualification, but only can give you that much confidence about the instrument. Is it working? That's it. So really, if you want to know whether the method is appropriate for the measurement of the sample, the product you are interested, they have to use the actual product, the actual material to perform the validation, dilution, right? Settings, all of those has to be using actual material. Okay, I think that concludes the first session. We're going to take just 3 slides. And again, thank you everyone for staying around till the last few minutes. And I see a lot of you participating in both the lecture, the comment response, as well as in the breakout room. I see a lot of your faces, and some group are quieter, some group are more, more lively. But nonetheless, I think everyone is being really, helping us to deliver this course, and then hopefully it's also useful for you. And this last bit of breakout room is something we're trying for the first time. So in case you feel that we are doing maybe too much or too little or not clear enough, yeah, bear with us. It's the first time we're trying. Hopefully, it's giving you some good flavor of challenges that we encounter every day during the development of the size, of course, analysis of the size and the particle size measurement, etc. So I thought to close the today by going back to the beginning when I mentioned on the first day, the measurement of the size is a problem that's not posed properly, or the question is a ill conceived problem. And what I was saying that the beginning that what we are truly interested is not the size. I guess that's a message I hope get across, that when you are looking at the size as a quality control tool, typically what you have in mind may be related to the stability of the product, may be related to the process capability, whether it's really resulting in blend uniformity, content uniformity, or maybe it's related to the dissolution, bioavailability. But why you are interested in knowing size and put a control on the size is because you care about what does the particle do. So in that regard, keep in mind size is only one of many factors that is more important. And also second part is that what we are actually measuring is not the size. Hopefully, you, by the example of the dynamic light scattering, where we're measuring the time-dependent phenomenon. By laser diffraction, we're measuring angular-dependent light scattering. What we are measuring is a response of particle with light, the interaction with the light. What we are not measuring is not the size itself, but we are converting the signal obtained from the scattering to something relatable to the size. Same thing goes to the AF4 fractionation, and also the imaging analysis for the subvisible particles. So in a way, what we are measuring may not be exactly the size. I thought to close this training with kind of recap. How do I want to do the recap is I'm going to describe what we have learned throughout the last few days in the theme of two. And the theme of two is a thing that I saw that repeatedly occurring, that just basically using that as a frame for us to kind of go through what we have learned throughout the last few days. The first thing is that we looked at both particles of desired and undesired particles. So we're looking at a particle that may be in the drug product, and also particle that may be subvisible, visible range that is undesired, and what are the technique we have been using to analyze this both type of particles. And also, as I mentioned, it's really important not only focus on the particle that we are analyzing, but also be aware of the environment, be aware of the medium that's used to disperse the particles, equally important because that impact the stability. And also two things, trueness versus precision, which one is easier to obtain, right? Precision is easier than trueness because what we consider as a true is really difficult to answer. And also whenever we are looking at some numbers provided by the company. Keep in mind that you have to ask question, what is the technique used to generate the number? So what is the equivalent spheres? And looking together with number together. The two things need to be together. You cannot looking at the number by itself without looking at what is the equivalent sphere, what is the technique, what's the underlying principle of the measurement. Even we are looking at the number, we need to consider two things. One is what's the average mean value, something about the average behavior of the particles. Also, we need to consider another number, which is a spread or the distribution of the particles. So whether it's polydispersity index or the distribution D10, D50, D90 in terms of the span value. So we need two sets of value in order to describe a fully particle size distribution. When we talk about light scattering phenomenon, there are two types of theory used to analyze it. One is Rayleigh scattering, the other is Mie scattering, which is related to two type of way to analyze data. One is time dependent, the other is, sorry, that's dynamic light scattering. I'm jumping ahead. We also looked at two different ways to analyze data. One is ensemble technique that like the DLS, like the laser diffraction, are ensemble. We're analyzing average behavior. And also we talk about the separation using AF4 to separate the particles before analyze them. And we talk about, yes, the dynamic light scattering and laser diffraction are the two most commonly used method, one for the nanomaterials and the other for all-purpose particles. Laser diffraction is more popular. But the two techniques, they're using different way to analyze scattering phenomenon. One is angular dependent and the other is time dependent, right? So the laser diffraction is angular dependent. We're measuring intensity as a function of angle. For the dynamic light scattering is a function of time. And in terms of the DLS, we're basically measuring two things. We're relying on two things. One is what the particle is moving in Brownian motion, and the other is when the particle is doing the Brownian motion, how do they interact with the light? So as a combination of motion and the interaction with light, we get the signal from fluctuation, which has two aspects to it. One, the light fluctuation can be constructive in nature, that resulting increasing the light signal, or can be destructive that they cancel out, so resulting in a decrease in the light signal. So that's what light is going up and down resulting in fluctuation. But that fluctuation is what allow us to obtain a correlation function G to able to calculate the diffusion and etc. But the G value itself depend on instrument as well as sample properties. And more on the correlation function, it has both a start and a decay. Starting point is when is earlier decay start, it's a smaller particles. If it's a later decay, then it's a larger particles. And also the slope of the decay actually related to the distribution. Certainly it's a smaller particles, we know they move faster. Larger particles, they move slower. So this is reflected in the fluctuation. And the DLS analysis, we talk about two-way analyzing it, cumulant and distribution. We talk about the dilution effect on the particle size can change the particle itself or change the environment, which resulting also the size value being changed. And for the DLS, there are two ways of analyzing by distribution data, general purpose, multi-narrow mode. What the difference is regularizer setting, basically multiple peaks. You can assume whether they're there, so apply different regularizer setting. And in terms of laser diffraction, smaller particles, remember, always scatter at a larger angle, diffraction at a larger angle, larger particles resulting in a smaller diffraction pattern. So that's it for all the classes that we have talked about. Of course, I emphasize more on the DLS laser diffraction because that's the two most commonly you will see in submissions. But here is again a slide showing all the contact. And I want to thank you for all the speakers, instructors, what's not on the screen, narration, Xing, who have helped us with a lot of preparation and also the breakout session. And certainly in the future, if you have any questions, please let us know. We recognize that this training can only provide a starting point, giving you some information, but it's really difficult to have digesting all those information in relatively short time. So please, if you have any question, let us know. and we are here to support you, and we are here to help you answer any question. And if you found some interesting examples, also let us know so that we can learn together with you as well. Lastly, thank you all for your time. I know it's very precious time for your review, a lot of things on your schedule. So appreciate you'll stay around for 3 days in the morning. And thank you for Janet for help us coordinating this course. And thank you, everyone.